and welcome to biology class. Friends, today we shall discuss about genomic imprinting. Before that, I request all of my friends, please subscribe my YouTube channel and watch, like and share my videos with your friends. A basic principle of Mendelian genetics is that the parental origin of a gene doesn't affect its expression and therefore reciprocal crosses give identical results. However, for some genetic characteristics, such as those are encoded by X-linked genes and cytoplasmic genes, reciprocal crosses do not provide same results. In these cases, males and females do not contribute the same genetic material to the offspring. With regard to autosomal genes, males and females contribute the same number of genes and maternal and paternal copies of a gene have long been assumed to have equal effect. However, in some cases, the expression of a gene is significantly affected by the parental origin. This differential expression of gene, depending on whether they are inherited from male or female parent, is called genomic imprinting. A gene that exhibits genomic imprinting in both mice and humans is IGF2 that encode a protein called insulin-like growth factor. Insulin-like growth factor 2. Offsprings inherit one allele of IGF2 from father and another allele from mother. The paternal copy of IGF2 is actively expressed in placenta and fetus, whereas the maternal copy remains completely silent. So in this case, the key to the expression of the gene such as IGF2 is the sex of the parent. IGF2 is expressed only when it is transmitted by male parents. In some other cases, genes are expressed only when they are inherited from female parents. Genomic imprinting has been implicated in several human disorders including Prader-Willi, and Angelman syndrome Many persons with Prader-Willi syndrome are missing a small region on the long arm of chromosome 15. The deletion of this region is always inherited from father. Thus, children with Prader-Willi syndrome lack a paternal copy of genes on the long arm of chromosome 15. The deletion of the same region of chromosome 15 can also be inherited. The deletion of the same region of chromosome 15 can also be inherited from mother. But this inheritance results in a completely different set of symptoms called as Angelman syndrome. Many imprinted genes are associated with many imprinted genes are associated with fetal growth. Genomic imprinting has also been reported in plants, for example, in the differential expression of maternal and paternal genes, in the endosperm, which, like the placenta of mammals, provide nutrition to the developing embryo. The mechanism of genomic imprinting is under investigation. However, methylation of DNA bases is essential to the process. Now the question is, why does genomic imprinting occurs? One possible answer is the genetic conflict hypothesis, which states that different and conflicting evolutionary pressure act on alleles of genes such as IGF2 that affect fetal growth. In humans, the IGF2 locus is on the short arm of chromosome 11, but in mice it is on chromosome 7. It is because of the different and conflicting evolutionary pressure, paternal allele of IGF2 is expressed and the maternal allele remain completely silent. The expression of paternal IGF2 stimulates the fetal growth. Because of the same evolutionary pressure, the maternal alleles are silenced, as a result of which the fetus is not grown beyond a limit. If both the alleles will be expressed, then the baby will be very large. Giving birth to very large baby is not helpful to the mother, since for this the mother has to contribute a large amount of nutrients that will limit her ability to reproduce in the future, and also because giving birth to very large baby is difficult and risky. So in humans, the size of the fetus is determined by the combined effect of paternal and maternal alleles of various genes. Friends, this is all about genomic imprinting. For more updates, subscribe my YouTube channel, Sam Biology Class.